Good morning, everyone. Today, we will begin one of the foundational blocks of general pathology. And in this session, sets the stage for much of what you will encounter throughout the rest of this course, and frankly, throughout your clinical training. General pathology focuses on the fundamental cellular and tissue responses that underlie all forms of diseases, regardless of the organ, system, or clinical presentation. Every disease process can ultimately be traced back to disturbances in cell structure and function. That is the central message of this lecture. Clinical disease is the outward expression of cellular derangement. When cells can no longer adapt, compensate, or maintain homeostasis, pathology emerges. We won't be discussing about chapter 1. Those are cells, the normal cells. You should have known that during your first year. But in the second chapter, we will be reviewing essential features of normal cell biology still, how healthy cells maintain their environmental environment, communicate, divide, and carry out specialized function. So this lecture will provide a baseline, what a cell should look like and how it should behave. Today, we build directly on the foundation. Our focus shifts from normal physiology to what happens when cells are challenged. When they have encountered stress, toxin, ischemia, infection, immune reaction, or genetic abnormalities. And we will explore how cells attempt to adapt, what happens when those adaptations fail, and the mechanism that leads to reversible injury. Irreversible injury and untimely cell death. So by the end of the session, you should have a clear conceptual map of how cells respond to injury and why these responses matter clinically. This understanding is essential because every disease you will study, from myocardial infarction to chronic lung disease to cancer, begins with the cellular events we are discussing today. Without further ado, I'll show you the content. So these are my contents. And these are my learning objective. And by the end of the session, students should be able to recall, distinguish, describe, and explain. So, cellular adaptation. Now, let's take a closer look at a major form of cellular adaptation. These are the strategies cells use to survive when they are exposed to increased demands, reduced nutrient, chronic irritation, or other stressors. Each adaptation has both physiological form, normal, expected response, and pathological forms, which occurs in disease state. Except for metaplasia, this is only what? Pathological. Actually, metaplasia has also physiologic, but there will be less physiologic but more pathologic for hyperplasia. So let's begin with hypertrophy. Hypertrophy refers to an increase in the size of individual cells, which leads to an increase in the size of the organ. This occurs in tissue that cannot readily divide, such as cardiac and skeletal muscle. A classic physiological example is the gravid uterus. During pregnancy, under the influence of estrogen, smooth muscle cells enlarge to accommodate the growing fetus. In contrast, a pathologic example is what? A cardiac hypertrophy. In long-standing hypertension, here the myocardium enlarges because it is forced to work against increased vascular resistance. But in both cases, the underlying driver include mechanical stress and growth factor signaling. But the outcome differ dramatically. One is adaptive and beneficial. The other is eventually contributes to heart failure. Next, hyperplasia, which is an increase in the number of cells, and this can only occur in tissue capable of cell division or those with abundant stem cells. A physiologic example is the proliferation of breast cyanide during lactation, driven by hormonal stimulation to meet the metabolic demands of breastfeeding. A pathologic counterpart is endometrial hyperplasia, which result from excessive or unopposed estrogen stimulation. So in this case, the increased cell proliferation can progress to dysplasia and even carcinoma if the hormonal imbalance persists. Now, moving to the next. This is atrophy, which is a decrease in cell size and ultimately organ size. Atrophy occurs when cells experience reduced workload, diminished blood supply, loss of innervation, inadequate nutrition, or aging. For example, disuse as atrophy occurs in immobilized limb while denervation atrophy affects the muscle after nerve injury. A key mechanism here is increased autophagy, or cells digest their own components to reduce demand and recycle nutrients. And atrophy is not simply shrinkage. It reflects a coordinated regulation of cellular metabolism. Finally, the next one, or the last one, is metaplasia, a reversible change in which one differentiated cell is replaced by another, better suited to withstand particular stress. And a classic example is squamous metaplasia in the bronchi of chronic smoker. The non-cellulated columnar epithelium is replaced by a tougher squamous epithelium that can be better tolerate smoke exposure. However, this adaptation comes at a loss, the loss of mucociliary clearance and an increased risk of infection and malignancy. Another important example is what? Barrett's esophagus, where chronic acid reflux induces transformation from squamous to intestinal type columnar epithelium. Again, the change is protective in the short term but increases the risk of adenocarcinoma. So key points across all this adaptation is that they are initially beneficial, allowing cells to survive in the face of stress. But when the stress is persistent or severe, this same adaptation can be maladaptive and set the stage for injury, dysfunction, and disease. Next, the mechanism of cell injury. This slide, I would like to use this slide as a frame the remarkable diversity of stimuli that can damage cells. Cells can be injured by a wide range of factors: oxygen deprivation, physical trauma, chemicals, infectious agent, immune reaction, genetic defects, and nutritional imbalances. Although these stimuli are very different from one another, they tend to converge on a limited number of vulnerable cellular targets. A key distinction to clarify early is the difference between hypoxia and ischemia, because these terms are often used interchangeably but represent different biological problems. Hypoxia refers to a reduction of oxygen availability. The blood flow may be still intact, but the oxygen content is insufficient. For example, in severe anemia or carbon monoxide poisoning. Ischemia, on the other hand, is a reduction of blood flow. This means not only the reduced oxygen delivery, but also reduced delivery of nutrients and impaired removal of metabolic waste. Because of this combination, ischemia is usually far more damaging than hypoxia alone. Once an injurious stimulus affects the cell, several downstream targets are particularly vulnerable. One of the earliest and most critical is the mitochondrial. Damage here impairs ATP production, leading to failure of energy-dependent processes. Another major target is the cell membrane. Both the plasma membrane and organelle membranes, loss of membrane integrity leads to leakage of enzyme, disruption of ion gradient, and ultimately cell death. DNA damage is another important pathway, especially in the context of radiation, toxins, and oxidative stress. And finally, disturbances in calcium homeostasis can activate destructive enzymes, including proteases, phospholipases, and endonucleases, accelerating cellular injury. To make this more concrete, let's anchor this mechanism in a simple clinical example. In a myocardial infarction, the underlying problem is ischemia and abrupt loss of blood flow to a region of the heart. The combination of oxygen deprivation, nutrient loss, and accumulation of toxic metabolites leads to rapid mitochondrial failure and irreversible cell death. In contrast, consider paracetamol acetaminophen overdose, which represents a chemical injury. The toxic metabolite NATQI accumulates in the liver, depletes glutathione, and causes oxidative damage to the mitochondria and membranes, ultimately leading to hepatocellular necrosis. This example illustrates that although the triggers differ, the pathway of injury often converge. Understanding this shared mechanism help explain why very different disease can produce similar patterns of cellular damage. Now, the reversible versus irreversible cell injury. I would like to highlight one of the most important concepts in general pathology, the idea of the point of no return. In cell injury, this is the threshold beyond which a cell cannot recover no matter how quickly or effectively the original stressor is removed. Understanding where the point lies and what marks the transition is essential for interpreting laboratory findings, imaging, and histologic changes in real clinical scenarios. So let's begin with reversible injury. Reversible injury represents a stage where the cell is under stress and its function are impaired, but its structural integrity is still largely preserved. The key idea is that these changes are functional derangement, not permanent damage. If the stress is relieved, if oxygen is restored, toxins are removed, or metabolic demands are reduced, the cell has the capacity to return to normal. Typical features include cellular swelling, fatty change, membrane bleeding, and detachment of ribosome. From the endoplasmic reticulum, these changes reflect energy, depletion, and ionic balance, but the cell core machinery remains intact. So in contrast, irreversible injury occurs when the damage surpasses the cell's ability to recover. So those two major events define this translation. First, the inability to restore mitochondrial function. Once mitochondria lose the capacity to generate ATP even after reperfusion or removal of stress, and the cell is committed to death without ATP, ion pumps fail, calcium accumulates, and destructive enzyme and becomes activated. Second, catastrophic membrane damage. This includes not only the plasma membrane, but also lysosomal and mitochondrial membranes. This lysosomal membrane ruptures, their enzymes spill into the cytoplasm and digest the cell from within. So when the plasma membrane fails, cellular contents leaks out and triggering inflammation and sealing the cell plate. If you have a histologic image available, this is an excellent moment to show them what? The nuclear changes that accompany reversible injury, like what? Pyknosis, cardiorexis, and cardiolysis. These three, I would like you to research what are these three. Also, highlight the increased cytoplasmic eosinophilia seen in necrotic cells. So this occurs because RNA is lost and denatured protein bind more strongly to eosin, giving the cytoplasm a pink or a bright pink appearance. This visual cue is essential for recognizing necrosis under the microscope. So together with this feature, the mitochondrial failure the membrane breakdown, the nuclear dissolution, and cytoplasmic eosinophilia, marks the irreversible stage of cell injury. Once these changes occur, the cell cannot recover. It is committed to necrosis or another form of regulated cell death. So understanding this progression helps you interpret everything from troponin levels in myocardial infarction to histologic findings in tissue biopsy. It is the bridge between cellular biology and clinical medicine. Now, let's turn our attention to necrosis, one of the major forms of cell death you will encounter throughout pathology. Necrosis refers to uncontrolled, unregulated cell death that results from severe or irreversible injury. So the defining feature is the loss of membrane integrity, both at the plasma membrane and within organelles. And once membrane fail, cellular content leaks into the extracellular space, triggering an inflammatory response. This is why necrosis is almost always associated with inflammation, unlike apoptosis. So within necrosis, we recognize several distinct morphologic patterns, each associated with particular clinical setting. So number one, coagulative necrosis, which is the most common pattern in solid organs, such as the heart, kidney, and kidney. Sorry. In coagulative necrosis, the overall architecture of the tissue is preserved for several days, even though the cells themselves are dead. This occurs because the injury denatures not only structural protein, but also enzymes that would normally digest the dead cell. And as a result, the tissue becomes firm, pale, and retain its outline. A classic example is a myocardial infarct. When a coronary artery becomes occluded, ischemia leads to rapid cell death, but the basic structure of myocardium remains recognizable for some time. The same pattern is seen in renal infarct, where wedge-shaped areas of pale, firm tissue reflects the vascular distribution of the affected artery. The second major pattern is liquefactive necrosis, which is essentially the opposite. Here, the dead tissue is completely digested, transforming into a soft liquid mass. It's like a river of dead cells. This pattern is driven by the action of hydrolytic enzyme, often released by leukocytes, and it is the hallmark of bacterial infection, where neutrophils flood the area and release. Enzymes that liquefy the tissue produce pus, and liquefactive necrosis is also characterized as in brain ischemia. As we told you, ischemia should be coagulative, but exception to the rule, the brain will be liquefactive. Unlike other organs, the brain lacks substantial support stroma. So when neural and glial cells die, tissue rapidly breaks down into a viscous fluid. And there are additional specialized forms of necrosis. You have the caseous necrosis, which is seen in granulomatous infection like tuberculosis, or caseous necrosis is seen in tuberculosis. Fat necrosis in trauma or in autodigestion of the pancreas. Fibrinoid necrosis seen in blood vessels, autoimmune blood vessels, each with its own clinical significance. And there's another necrosis, we call it gangrenous necrosis. But this is not used in pathology, but used clinically. This is a combination of coagulative and liquefactive necrosis. So next, our focus now is in apoptosis, a fundamentally different form of cell death from necrosis. It's important to contrast the two clearly because they arise in different contexts and have a very different consequences for tissues. Unlike necrosis, which is uncontrolled, messy, and inflammatory, apoptosis is tightly regulated, energy-dependent process. It allows cells to die in a controlled manner without spilling their contents or provoking inflammation. In many cases, apoptosis is not the only normal but essential for healthy tissue function. It is the body's way of removing cells that are no longer needed and are potentially harmful or have sustained irreparable damage. Apoptosis can be triggered in two major pathways, an intrinsic and extrinsic pathway. Intrinsic pathway is also known as the mitochondrial pathway. Is regulated by the BCL2 family of protein. This protein acts as molecular switches that determines whether the cell will live or die. When a cell experiences severe DNA damage, misfolded proteins, or growth factor deprivation, pro-apoptotic members of the BCL2 family promote mitochondrial outer membrane permeabilization. This releases cytochrome C in the cytosol, which activates caspases, the enzyme that executes the apoptotic program, and the anti-apoptotic protein like BCL2 or BCLXL oppose this process, and the balance between this group determines the cell's fate. The extrinsic pathway, on the other hand, is initiated at the cell surface through that receptor, such as the FAST receptor and the TNF receptor family. When this receptor binds their ligands, they recruit adapter proteins that activate caspases directly, and this pathway is especially important in immune regulation, including the elimination of harmful or unnecessary lymphocytes. To appreciate the importance of apoptosis, consider some key physiologic examples like during embryogenesis. Apoptosis stops developing tissue, for instance, removing the webbing between the digits. In hormone-dependent involution, such as the regression of the endometrium during menstruation or the involution of the lactating breast after weaning, apoptosis ensures orderly tissue remodeling in the immune system. Apoptosis eliminates self-reactive lymphocyte, preventing autoimmunity, and when cells accumulate irreparable DNA damage, apoptosis prevents them from becoming malignant. However, apoptosis is not always beneficial. When it becomes dysregulated, it contributes to a wide range of disease. Insufficient apoptosis allows damaged or mutated cells to survive, contributing to cancer and certain autoimmune disorder. Excessive apoptosis, on the other hand, plays a role in neurodegenerative disease, the ischemic injury, and some viral infection. So while apoptosis is often physiologic and protective, its balance is too critical. Too little, too much can drive disease. Understanding this pathway helps explain why certain therapies such as BCL2 inhibitors in oncology targets the apoptotic machinery directly. So, in the final section of today's session, I would like to introduce you an important idea. When cells are exposed to chronic stress or ongoing injury, they don't die immediately. Instead, they often undergo long-term adaptive changes. And one of the most common outcomes is the accumulation of various substances within the cell. So this accumulation can be harmless, reversible, or profoundly damaging depending on the context. So let's begin with some major categories. One of the most familiar is steatosis, or fatty change. This occurs when triglycerides accumulate within the parenchymal cells, most commonly in the liver, and it can result from alcohol use, obesity, diabetes, or toxins that impair lipid metabolism. And under the microscope, you'll see clear vacuoles displacing the nucleus, a hallmark of fatty liver disease. Another important category is cholesterol accumulation. This is especially relevant in atherosclerosis, where macrophage in the arterial wall becomes loaded with cholesterol esters, forming the classic foam cells that contributes to plaque formation. We also see protein accumulation, such as reabsorbing droplets in the proximal renal tubules during proteinuria. These droplets represent that the cell attempts to reclaim filtered protein, but when the load is excessive, the protein accumulates faster than they can be processed. Glycogen accumulation occurs in metabolic disorder like diabetes or glycogen storage diseases, where impaired regulation leads to excessive intracellular glycogen deposit. And there are pigments, which can be either exogenous or endogenous. A key endogenous pigment is the lipofuscin, often called the wear and tear pigment. It accumulates slowly over time as a byproduct of lipid peroxidation and is a marker of aging or chronic oxidative stress. Another is hemosiderin, an important iron-containing pigment that accumulates in condition of excess iron, such as blood transfusion or chronic hemolysis. Beyond this accumulation, chronic injury can also lead to pathologic calcification, and which comes into two major forms. Dystrophic calcification occurs in dead or dying tissues, areas of necrosis, damaged valve, or atherosclerotic plaque. Importantly, serum calcium levels are normal. The calcification reflects local tissue injury, not systemic imbalance. In contrast, metastatic calcification results from hypercalcemia, often due to hyperparathyroidism, renal failure, or vitamin D disorder, or bone destruction. Here, calcium deposits on otherwise normal tissues such as kidney, lung, and gastric mucosa. And finally, cellular aging, which represents the cumulative effect of lifelong stress, metabolic activity, and imperfect repair mechanism. So several key themes define this process. First is telomere shortening, which each cell division telomere become progressively shorter. When they reach a critical length, the cell cannot replicate senescence and can no longer divide. Second, accumulation of DNA damage. Even with robust repair system, accumulative oxidative stress and environmental exposure gradually introduce mutation and structural damage that impair cellular function. Third, defective protein homeostasis. Aging cells show reduced efficiency and chaperone-mediated folding and diminish proteasomal and autophagic degradation. Misfolded protein accumulate contributing to degenerative diseases. And finally, altered nutrient sensing, particularly through pathway involving IGF-1 and sirtuins. Reduced IGF-1 signaling and increased sirtuin activity has been linked to enhanced longevity in model organisms, largely through improved metabolic efficiency and DNA repair. And together, these processes, intracellular accumulation, calcification, and the molecular mechanism of aging, represents the long-term consequence of stress and injury. They remind us pathology is not only about acute events, but like necrosis or apoptosis, but also about slow, cumulative changes that shape tissue function over a lifetime. So putting it all together, this is a conceptual map to bring everything together. We've covered adaptation, injury, cell death, and long-term cellular changes. Now let's integrate these ideas into a single coherent clinical scenario. Let's consider a patient with a long-standing hypertension. This is an excellent example because it illustrates full spectrum of cellular responses that we have discussed a while ago. So in the early stages, the myocardium responds to the increased workload through hypertrophy. This is an adaptive response. The cardiac muscle fibers enlarge to generate one force and maintain cardiac output. At this point, the process is still physiologic in intent. Even though the underlying stimulus is pathologic, the heart is compensating. But over time, as the hypertension persists, the adaptive capacity of the myocardium becomes exhausted, and the increased wall thickness leads to higher oxygen demand, yet the coronary arteries cannot proportionally increase their supply. So this mismatch sets the stage for ischemia. Here I want you to identify the transition point where the adaptation and an injury begins. So this is the moment when the cell compensatory mechanism can no longer maintain homeostasis. So if the ischemia becomes severe or prolonged, the affected myocardial cells undergo irreversible injury leading to coagulative necrosis, a myocardial infarction, the loss of membrane integrity, mitochondria failure, and nuclear breakdown mark the point of no return. This is where the mechanism we discussed, ATP depletion, calcium impact, free radical damage, becomes dominant. Following the infarction, the body initiates a healing response, necrotic tissue or removed by inflammatory cells. And over time, the area is replaced by scar tissue. The fibrotic repair restores structural integrity but not contractile function. So it is a long-term consequence, death, and represents the culmination of the entire sequence, adaptation to injury to death to repair. So as you see this scenario, I want you to think critically about the stage of each stage. Which mechanism dominate during which part sorry which mechanism dominate during hypertrophy and which pathway are activated during ischemia? What marks the transition to irreversible injury and how does the tissue respond to necrosis? These questions are not just academic, they are the basis of understanding clinical presentation, interpreting laboratory results, and predicting patient outcomes. And finally, I encourage you to start linking this framework to the topics that we'll be exploring next. Inflammatory response, the acute and chronic, the proliferative signals involving hypertrophy, repair connect to neoplasia. So this conceptual map is an anchor if you can follow a single clinical scenario. Through this stages, you are well on your way to master now the logic of patho. Any questions? So if there's no question, thank you. It's not about how hard you hit. It's about how hard you can get hit and keep moving forward. Rocky Balboa. This is from Rocky Balboa. It's not only a life quote, but it's like what? How our bodies survive. It's not about how they are being hit, but how they get up. So these are my references.